So we were interested in whether we can identify things that may predict whether somebody will respond as well as possible to an IMiD drug, so lenalidomide and pomalidomide, or not. Because at the moment we treat everyone and we wait to see whether they'll respond or not, and there may be a more efficient way of identifying up front the patients who will and may not respond. And the way in which we've been addressing this is through looking at function of the protein cereblon. Cereblon is the obligate binding partner of all the IMiD drugs and the new CELMoD drugs as well, so it's extremely important, essential for these drugs to work. And what we found last year was that actually as patients progress through becoming sequentially refractory to one IMiD drug and then another, up to a third of them lose this protein because they have a genetic aberration which causes it to fall. And because that was so interesting and important, we then went on to try and identify whether there may be other genetic causes of loss of cereblon and therefore acquisition of resistance to IMiD drugs. And we looked across the whole genome, but we looked particularly at genes that have been identified in vitro already by CRISPR screening methods. And there are some really beautiful papers that have looked at this in the laboratory, but it's never been looked at in patients. So what we did is we identified the genes that had been shown to be essential for responding to IMiD in the lab, and we looked at that set of genes in patients. And there were just over 20 genes like that from the initial in vitro studies, and we looked at all of them in a big patient data set, which had cohorts of patients that had never seen IMiD drugs before, ones that were resistant or refractory to lenalidomide, and then a third cohort that were refractory to pomalidomide. And we identified one region of the genome, chromosome 2q, which is sequentially lost as patients become refractory to these drugs, just in the same way that we'd previously seen with the cereblon region, which is on 3p. So this region 2q contains two genes that are part of a complex called the COP9 signalosome. And the COP9 signalosome has many roles, but one of its role is in maintaining homeostasis in the activity of the cereblon E3 ligase. And when you lose members of the COP9 signalosome, actually what happens is that E3 ligase is switched on all the time, and cereblon itself becomes auto-ubiquitinated, which means it gets destroyed. So when members of the COP9 signalosome are lost, the feedback therefore is that cereblon itself becomes destroyed, and that is a mechanism by which patients may respond less well to the drugs, which require cereblon to work. So we found that as patients became resistant to these drugs, up to about 15% of them lost this region. And then in a separate set of patients, we were able to track this happening with sequential samples from their bone marrow as they became resistant through these sequential drugs. And we were able to capture that subclone of the 2q loss and track its expansion till it became clonal at the time they were drug resistant. So I think why this might be important is that along with the previous work we did on the loss of cereblon on 3p, is that they're both mechanisms that show that certain genetic traits are being selected as patients become drug resistant, to the point where when patients are resistant, there are certain genetics which point out that actually they may now be resistant to this class of drugs. And we can monitor for these really easy because they're copy loss, copy number changes, which can be picked up with targeted genetic tests. And this is a really potentially simple way that precision medicine might be used to guide our choice of therapies in myeloma when we have such a lot to choose from.